Today, part of what we will be talking about is our advanced genomic services. We have a dedicated innovation and development team who work on cutting-edge technologies, which help deliver to you new powerful services, such as long-read microprofiling, single-cell technologies, and 3D genomics. We also have access to a large suite of infrastructure, including the largest NovaSeq sequences, also long-read sequencing through the PacBio Revio and the Oxford Nanopore Promethean series. Today, we're going to be talking on plant pangenomes. Plant pangenomes have followed on from strong advances in generating comprehensive reference genomes for significant food crops across the world, such as rice, maize, wheat, legumes, and several horticultural crops. Pangenome assembly integrates data from multiple individuals to construct a comprehensive genomic landscape, revealing both core and accessory variable genomic elements. Additionally, Hi-C data provides long-range information about these genomes, and hence pangenomes that allow chromosomal scale assembly and identify long-range structural linkages of functional genomic variants. Pangenomes and Hi-C data are particularly useful for crop improvement, as they can help identify genetic variants associated with desirable traits such as yield, drought tolerance, and disease resistance. Pangenomes can also be used to support crop improvement through the identification of genes lost from the germplasm through domestication. AGRF is working with Professor Rajiv Vaishni's group at Murdoch University in Perth on a series of pangene projects for grain and horticultural crops. In the past year, we have provided both long-read sequence data using the PacBio Revio system and Hi-C data using the OmniC protocol from Dovetail to include in the assembly of pangenomes for wheat, banana, passion fruit, custard apple, and pineapple. Today, we have two talks on the technologies used in this area. Firstly, James Miller from PacBio Systems will give his talk on chemistry of PacBio. Then Dr. Danya Suraj will talk more about Hi-C and pangenomes in particular. Firstly, I'd like to welcome James Miller, Regional Commercial Lead of ANZ PacBio, to talk about PacBio technologies. Over to you, James.
Thanks very much, David. And I'd like to firstly just thank AGRF for hosting this webinar and inviting me to speak today. So I'm going to talk about the latest Hi-Fi applications in plant and animal research today. But initially, first, I'm just going to give just a brief review about what Hi-Fi reads are, PacBio's primary data type. So Hi-Fi reads, to generate Hi-Fi reads, or Hi-Fi reads are single molecule real-time sequences that we generate by anchoring a polymerase molecule and a DNA molecule to the base of a little chamber, and we feed it fluorescently labeled nucleotides, and we record that information. But what we're able to do is to sequence that molecule multiple times, and from those multiple reads, we build up a highly accurate consensus read, and that's what a Hi-Fi read is. They're 90% of the reads coming off our sequences are Q30 quality or above, and up to 25 kilobases in length. And the sequencing is done on these consumables called a SMRT Cell, where there's millions of these molecules being sequenced in at the same time. So this data type produces long reads, as I said, up to 25 kb, very, very accurate. We get complete coverage of a genome, and we can also sequence the epigenome at the same time. Now, Hi-Fi sequencing can be done on two different platforms at the moment. We have a benchtop platform we released late last year, and that can run up to about 200 genomes per year, one SMRT Cell and at a time, and 60 gigabases of Hi-Fi data per SMRT Cell per run. We also have a higher throughput Revio system, which has just had Spark chemistry released on it, a new chemistry late last year, and that will achieve about 2,500 human genomes per year and up to 480 gigabases per run. And this is the system that AGRF are running as a service. Importantly, we also lowered the input for the Revio with the new Spark chemistry. So this is just a brief snapshot of this Spark chemistry, which was released late last year. It allows us lower input down to 500 nanograms of DNA per library, and we increased the output of the Revio for the same amount of cost per run by about 33% compared to the previous chemistry. So it's a really major upgrade for us. And that enables actually on the Revio system on one SMRT Cell for you to run up to 2 genomes in one SMRT Cell, or more of what you previously may have been doing per SMRT Cell. So it's just lowering the cost of sequencing. So now I'm going to shift and talk about applications that are relevant for plant and animal science. Brief summary of reference genomes, as well as low-pass sequencing application that we're really excited about, as well as enabling low input samples from a new amplification protocol that we have, which is also enabling long-read high-C application. So as many of you are aware, PacBio is the preferred technology for genome assembly. Here's a publication which analysed thousands of genome assemblies showing the contig N50 length, showing that HiFi really shines and produces more complete assemblies, and the highly accurate HiFi reads make those assemblies really accurate, which is a nice statement from this paper calling for greater uptake of highly accurate reads in future studies. Many of you will be aware that PacBio is the core technology for many genome initiatives, including Darwin Tree of Life, Earth Biogenome Project, and many published, many high-quality genomes that have been published. Dania is also going to talk about in more detail about pangenomes, but HiFi is now being used more and more commonly to produce pangenomes. A couple of examples here of the maize and soybean pangenomes showing discovery of SVs, and many of these SVs are driving phenotypes. I also want to call out as well with these projects, many people are using our PanDNA kits. We released a nanobind kit called PanDNA, which enables lots of different sample types now. We just have one kit for extracting DNA, and it's very important to produce high quality DNA if you're going to have a high quality genome assembly, and these kits are obviously compatible with plants as well. So we recommend using these kits for extraction of your DNA. Next thing I'm going to talk about is demonstrating the power of HiFi low pass for whole genome sequencing. So there's been a bit of an effort within PacBio, as well as our customers, to look at low coverage genotyping by sequencing type applications to be able to get at structural variants and see how well long reads perform in low pass genotyping by sequencing type applications. And this is an example from wheat where just as low as 0.3x to 6x coverage of HiFi produced very high precision calls of SVs. So we know that low coverage of HiFi is very powerful in genotyping by sequencing applications. I'd love to talk to any of you about that if you're interested. Here's another example in peanut where Josh Clevenger and Kendall Lee's group, Kendall pictured here, have been working with peanut low-pass genotyping by sequencing, where they show that the long reads in comparison to short reads map with much more confidence back to the genome, and they're able to cover more of the genome, 55% of the genome, compared to just 17% with short reads, showing you access more of the genome with long reads. And again, they're able to recover very high quality genotype calls with long reads showing many more SNP genotype calls being recovered compared to short reads after filtering, and ultimately showing an 8.5 decrease in cost per value compared to short reads. So we're pretty excited about this application, as well as discovery type things you can do with this application, where, for example, here they're able to identify R genes or resistance genes within the data, within the long reads, the short reads completely missed. And also we have released a high throughput library prep kit to enable these types of high throughput applications where you're making lots of libraries and multiplexing them on a single smart cell, where it's lower cost to produce these libraries. Now, the last thing I wanted to talk about is Amplify, which is an amplification protocol that we have just released in the last month or so, which enables sequencing from very low amounts of DNA down to 50 nanograms of less DNA, and also enabling applications like long read high C with a community developed sci-fi application. So this application uses amplification to try to amplify fairly large molecules. You can see up to about 7.5 or even a tail out to 15 kb of molecules. And these libraries also have very nice even coverage across the genome with the enzymes that we're using. As you can see here, pretty even across all of the GC content of the genome. So we get pretty good yields from these types of libraries, and we can make libraries from down as little as 1 nanogram of sample. This is a human genome example. And you can even assemble that data where you can produce haplotype resolved assemblies. You can see here you've got a haplotype resolved assembly with a reasonable contig N50 of the haplotype resolved assembly. So this enables you to sequence from very low amounts of sample or low quality sample and still be able to produce nice HiFi genomes. The other thing that this Amplify protocol is enabling is a community-developed protocol called Sci-Fi. And Sci-Fi is essentially a long-read high-C protocol, which leverages the same proximity ligation libraries that you would, or samples that you would produce through a standard 3C protocol. But then we go into the Amplify protocol, sequence on Revio or Vega, and then use some of the existing community tools for data processing. But what that enables is a single read from these libraries enables you to identify multiple different contact points and basically a richer amount of data for proximity ligation. Sci-Fi also improves the ability to map interactions across more of the genome, and you can produce haplotype phase chromosome scale assemblies here. You can see a haplotype resolved contact map here from the Sci-Fi data because the long reads are longer and they map more accurately to the genome, as well as the long reads being able to access more of the genome here. You can see the site where short reads fall down and you've got no coverage. You have nice coverage with the long reads. So we're pretty excited about this particular long read high-C application. So in summary, Hi-Fi really is the preferred technology for genome assembly. Low-pass sequencing applications we're pretty excited about. Hi-Fi is delivering significant value from the work that's been done on it thus far. We're enabling low input samples with our Amplify protocol, and our Amplify protocol is also enabling a long read high-C, which I think is a pretty exciting application. So thanks very much, and with that, I'll hand back to David.
Thanks, James. That was an excellent talk. Now we're going to move on to Danya Suraj.
Thank you, David, for a great introduction. So in my talk today, I'll briefly introduce the concept of pangenome, and then I'll talk about process of generating a chromosomal scale reference assembly using OmniC. And finally, I present a case study using wheat as a model and how we generated both Hi-Fi and Hi-C data to generate the plant pangenome here. So why is it important to build the pangenome in plant research? As we've seen from the previous presentations, in the recent years, there's been a surge of studies focusing on genomic sequencing of plants and comparison of multiple related species. A high degree of genomic diversity led to the realization that a single reference genome does not represent genomic diversity within a species, hence led to the concept of pangenome. So pangenome represents the genomic diversity and it has been annotated as a single file, which contains both core and variable regions. To illustrate this further, I have a Venn diagram on the left side, which is showing the genomic variations within a species, but in different lines of crops. So as you can see, there are areas of genome which are unique for certain individuals, or there are regions which are called dispensable, which are shared between different individuals within the species. Additionally, there are regions of the genome which are present in all cultivars or lines of crops are called core genomes. The unique regions and the dispensable regions are collectively called variable regions. And these regions of genomes are shown from previous studies that are highly enriched for genes regulating biotic and abiotic stresses, hence of a great research interest. Initially, the pangenome concept came from studies in bacteria in the year of 2004-2005. But then it took almost 10 years to generate and publish the first pangenome in soybean. As you can see, the graph, the bar graph on the right, ever since the number of publications came out from the plant research is steeply increasing and it's keep growing. What are the two different approaches used in plant science to build the pangenome? So the first method is a traditional or de novo assembly, and the second method is an iterative assembly or reference guided assembly. Multiple genomes of multiple individuals are assembled and then compared when it comes to de novo assembly. As you can see from the left in this cartoon, genomes of 3 different representation of a species has been built. The area which is commonly shared between these individuals are highlighted in blue, which are referred as core, as we said in the previous slides, and the regions which are unique in certain individuals are highlighted in orange, or or areas which are commonly present between more than one individuals are called dispensable regions. The iterative assembly all starts with a single reference genome. Non-redundant sequences from other individuals are sequentially added up to this reference genome to build the pan-genome reference. Unaligned reads are assembled together to make a new condig. Both approaches has its pros and cons, but iterative assembly struggles when it comes to complex regions of the genome, such as repeat regions. So for today's talk, we'll focus more on the de novo assembly formalities. Once built, the plant genome is a comprehensive atlas of gene content of a species, and it's a catalog of structural variants, copy number variation, present-absent variation, and single nucleotide polymorphism. What are the applications of a great reference? As the slide says, the best genome grows the best crops. So high quality assemblies of agriculturally important plant and insect pests are equally important. Help us to identify the genes and pathways linked to the desirable traits, such as drought tolerance, disease resistant, and crop improvement. They also help us to understand if the structural variation, such as deletion or insertion, is affecting the adaptability of the crop, or mixing of genes such as hybridization, generating a new variety of the crop which is having improved traits, or a specific allelic expression of the gene variant expression is regulating or providing the plant benefit during special conditions. In addition to that, it's a great tool for understanding polyploid or organism with more than 2 sets of chromosomes. In plant, majority of them are. And it also helps us to accurately classify the plants and understand the evolutionary relationship within the species and associated species. It's a great inventory for genetic markers. So enhanced breeding program and management effectively carried out using a better genome assembly and enabling us to control the pest and manage the pest management strategies. What are the processes involved in genome assembly? It all starts with generating a skeletal of draft assembly, if you like. And it starts with generating hi-fi reads using the PacBio platform. Hi-FiSM is the algorithm used for annotation and analysis. Once built, it generates long stretch of DNA, which now called as condigs. These condigs data, along with the Hi-C data, in our case it's Omni-C data, combined using scaffolding pipeline generates chromosomal scale assemblies. Cell-set2 or 3D DNA are generally used free softwares which are used for scaffolding. Once finished, once completing this scaffolding, one should have a chromosomal level contiguous assembly of that organism, which is ready for annotating. Using mRNA data generated from the same organism, allocate the physical positions of each genes, and now once completed, it is ready for publishing the genome. And at this stage, one should have a highly accurate, high quality and highly contiguous assembly with high confidence can be published at this stage. At AGRF, we provide services such as our dedicated PacBio team helps to generate the condig assembly or the draft assembly, and the advanced genomic team takes care of the Hi-C data, and we have NGS team who will be able to do all the NGS like mRNA and DNA sequencing, and our bio team helps to generate data for catered for the specific requirements of the clients. When we look at the molecular features of genome assembly, it does require information captured from DNA in its linearized or 2D structure needs to be combined with the information captured on its 3D structure or interactions from its information captured from the proximal interactions. Third generation sequencers such as PacBio or ONT are generally used to generate the condig assembly or the scaffold assembly. As James explained, because of its high base per accuracy and the capability of better handling the repeats or complex region, PacBio is advised to, in many cases to build better condig assemblies. And additionally, it generates more contiguous and complete assembly with lower computational needs. And these platforms generates condigs which are stretching up to kilobases. But we know that the chromosomes are megabases. So there comes the high-C data, which generates the chromosomal scale scaffold. Using the high-C data, we can order and orient these condigs generated from the previous platforms and then generate a chromosomal scale assembly. Proximity ligation technology, or otherwise known as high-C, generates chromosomal scale scaffold. It captures the physical interaction between the regions of the chromatin while they are in 3D structure, but these regions are far distant in their linear coordinate. Or in other words, proximity ligation reaction brings the informations together between the regions which are far apart in the chromatin, which is impossible with other technologies. So generally, the proximity ligation is carried out using restriction enzyme. This can be one enzyme or cocktail of enzymes. Because of its sequencing dependency, it generates uneven coverage, hence require a lot more sequencing to cover the genome. Last year, we established a partnership with Dovetail Genomics, who are the gold standard providers of the high-C technology to boost the innovation across Australia and New Zealand. In fact, we are the only certified provider for the APAC region for providing Omni-C technology. Omni-C using DNase-1, which is a sequence-independent kinetic enzyme. It has uniform access across the genome, hence generate shotgun-like sequencing coverage while capturing the long-range data. When we look at the molecular events, we can use cells, tissue, or organism as an input material for this. In the event of agriculture or plants, we use young leaves. These leaves are not we are not extracting the DNA from the material. In fact, we do an in situ chromatin cross-linking, which stabilizes the scaffold. And then we use the DNase digestion, DNase enzyme, DNase-1 enzyme for enzymatic digestion of the chromatin, which then followed by the end repair of this chromatin. And then the ligation reaction is added, which randomly ligate the DNA fragments which are in close proximity in their 3D interactions. These ligation events will provide a distribution of proximity ligation events ranging from kilobases to megabases. These chimera molecules which are produced from this ligation event then reverse cross-linked and libraries are generated using the next NGS library prep. Let me explain how high-C data is required for ordering and making the orientation of the chromosomes and generating the chromosomal scale assemblies. For example, these are the condig assembly which is coming out of the PacBio, if you like, or the draft assembly. So there are 4 condigs, condig A, B, C, and D. And then if we look at the interaction between these condigs, there's a strong interaction between condig A and condig C compared to condig A and condig B. There's no interaction or fewer interaction between condig A and condig D. Hence, the order has to be condig A, then condig C, and condig B, followed by condig D. For the same high-C data, if you look at the orientation of the interactions, the right side of condig A is strongly associated or have a higher, stronger interactions with condig C, right side of the condig C, and the right side of the condig A is lesser interactions with the left side of the condig C. So the condig C should be facing condig A and then followed by condig C. So this is how the chromosomal scale scaffold has been generated. So if we look at the bigger picture, a species or an organism with thousands of condigs will go through the proximity generated from the draft assembly, then combined with the proximity data generate chromosomal scale assemblies. The one way of looking at these interactions of the is generating the contact plot, otherwise known as link density histograms. As you can see on my left, there's hundreds and thousands of smaller squares are organized on the diagonal axis here. These data are representing the condigs which are generated from the draft assembly. At this point, these data doesn't make much of a sense here. But when combined with the proximity reads using scaffolding pipeline, it then generates something like that on my something like that. This contact plot on my right side. Here, it's far lesser condigs. Each boxes are organized and separated well. Each of these boxes are representing the chromosomes of that species. And if we count the number of chromosomes, that represents the total number of boxes will represent the number of chromosomes for that particular organism. So now we established why we require both Hi-C and Hi-Fi data to generate chromosomal scale assemblies for any species. So I'll move on to the case studies. Case study now. Earlier last year, we embarked on an exciting collaboration with Professor Rajiv Boshni from Murdoch University. One of the project was looking at building pangenome for bread wheat. If we look at the genomic features of the bread wheat or the wheat itself, it's a hexaploid species, which means it has inherited 6 sets of chromosomes from 3 ancestral species. So it has 7 chromosomes and 3 genomes and 2 copies of totally making 42 chromosomes. The average total genome size is around 17 GB for wheat, and it's more than 80% of the genomes are repetitive regions made of transposable elements. As you see from here, around 107K has high confidence genes has been identified across 3 genome sets in the wheat. So this is a complex build. Being hexaploid, wheat is a complex build, being hexaploidy with high, bigger size genome, and the complexity associated with the 6 set of chromosomes and with the repeat regions. So what we did here at AGRF, our PacBio team, since James has gone through a lot of the chemistry details, I'm not going through the chemistry, but briefly, the high molecular weight is, DNA is extracted from the plant, and then it has gone through the standard library prep where shearing has been done to generate the required size, and then adapter ligation and cleanup, and then Hi-Fi library is generated and then sequenced. I would like your attention around the BESCO complete contiguity score here. So completeness is around 99.7%, indicating that this is a really good assembly. And the 0.3% is either fragmented or missing BESCO score, which is showing very less amount is what is missing in this assembly when it comes to completeness. And when we look at the number of condigs, we generated 4,896 condigs totally, and with no surprise, 97% or almost 98% is duplicated condigs. So at the same time, advanced genomic team at the AGRF was making OmniSeq data out of the wheat, where the DNA is not extracted. In fact, we are performing in situ chromatin cross-linking and then digestion. One important step, one important highlight of this chemistry I wanted to highlight here is the inbuilt QC step. It has inbuilt QC step in every critical step of the workflow. And our library prep also include a low-pass sequencing, which analyze the percentage of valid read pairs and the complexity of the library before going into the deeper sequencing. So once completed, we then look at how Hi-C data is helping polishing this hi-fi data or the Kandig assembly. The one thing I wanted to tell here is like for wheat, because the number of libraries required to do the scaffolding varies, depends on the size of the genome and also the complexity. Here we are only using 2 times data to show that this Hi-C data is polishing the hi-fi data or the Kandig assembly, right? So 78,000 or 78 million read pairs, usable read pairs are used for this analysis, which is equivalent to 2 times coverage of 17 GB of the total genome of wheat. So as you look at it, the largest scaffold for the input assembly, in this case, hi-fi assembly, which we used for this specific cultivar, it's around 3 GB. But when using 2 times data, it's already generated 7.2 gig largest scaffold. We were able to join 638 gaps within the Kandig assembly, and the number of scaffolds are reduced from 3,703 to 3,068. So as you can see from my left, the contact plot is still in the format of highly contiguous. But what I wanted to highlight here is, if you look at these 2 boxes here, which is equivalent to chromosome 2 and 4, is already generating chromosomal scale assembly, as it is been getting tightly separated from other. So with 2 times data, it's already generating chromosomal chromosomal scale assemblies. Finally, I wanted to list some of the services we provide under advanced genomic, which would be essential for the crop science and agriculture, such as OmniSeed, prep, and microseed. Depends on 3D or epigenetic informations we wanted to capture from the plant science. We also keen to develop the current run and attack-seq for the plant. Finally, I wanted to acknowledge our partners and the teams involved in generating this data. And thank you, everyone. Thanks for listening and tuning into these presentations.